In our last lecture, we talked about mitosis, which is how all of the cells in our body are going to replicate themselves and divide. Now we're going to take a look at meiosis, which is a similar process, but these occur in the sex cells of a organism. So this is how an organism is going to, replicate itself and be the basis of sexual reproduction. After sexual reproduction, the offsprings are going to receive a unique combination of genes from the two parents. Okay, and so these combined set of genes is going to be able to program the offspring in a unique combination of traits. So we call this homologous chromosomes. So these are chromosomes that are a matching pair. You get one from mom, you get one from dad. That's what we call a homologous chromosome. So in us humans, we have in our somatic cells, so that's every other type of cell in our body besides our sex cells, besides the eggs and the sperm. In the humans, we have 46 chromosomes, or 23 pairs, and we can see that over here in what's called a karyotype. Okay, so when we display it like this in picture form, is called a karyotype, and we can see that we have 23 pairs. We have 22 pairs of what we call autosomes and one pair of sex chromosomes. So the autosomes are going to control all of the other features, in our bodies, and the sex chromosomes are going to be what determines whether we are male or female, and it also has some other features on there as well. So the homologous chromosomes, the one we get from, one that we get from our dad and one we get from our mom, are going to carry the same sequences of genes that control the same inherited characteristics. So, if chromosome number 1, has eye color, hair color, freckles, and stuff like that from mom, chromosome number 1 from dad also has those features as well, or either controls, having freckles or not, or what color hair and eyes and stuff like that. Now, two genes at a specific place on the chromosome may be slightly different versions, like I say. So, for example, my kids have a, blue-eyed, basically, gene from me and a brown-eyed gene from their father. So, we've got, slightly different versions of those genes, but they are on, the same homologous chromosomes as each other. And as I mentioned before, the sex chromosomes are going to determine the organism's gender. So, if you are female, if you have 2 X chromosomes, one from mom, one from dad. You are male if you get your X from mom and a Y chromosome from dad. So the picture over to our right, this karyotype, is of, since it's an X and a Y, this would be a male organism. So we talk about the somatic cells of our body. They are what we call diploid cells. Okay, they have 2 sets of those 23 chromosomes, whereas the sex cells, the gametes, which are the eggs and the sperm, have a single set of 23. So we call those haploid. And we can, kind of imagine if the sex cells weren't haploid, or half of the chromosomes, then we'd be doubling the amount of chromosomes in each generation afterwards. So, humans, the first humans may have started out with, 23 pairs, and then we'd get 46 pairs, and then 100 and, I can't even do math, but, keep going on and on. So it's basically, the way for the organisms to make sure that they have the same amount of chromosomes generation after generation. So our somatic cells, the ones that are, in every other part of our body besides our eggs and sperm, are what we call diploid. They have the 2 sets, one set from mom, one set from dad. And our sex cells are going to be haploid, or half that. They're going to have just one of the chromosomes of each of the 23. Alright, so when a sperm unites with an egg at fertilization, it forms a zygote. Okay, remember, the sperm cell is haploid and the egg cell is haploid, and when those two come together, we get a diploid zygote. So like I said before, we have to produce these haploid gametes so that we keep the chromosome numbers correct generation after generation. Otherwise, we'd start doubling, and then, by the time fifth generation comes around, we've got, hundreds and hundreds of chromosomes in each cell, and that would just make it kind of go haywire. So we've got to have a process of getting half of the chromosomes in eggs and sperm versus, all 23 sets in the somatic cells. Okay, so we need to be able to divide that set in half and put it in our eggs and our sperm. And so that process is what we call meiosis. It's cell division where chromosome number is reduced to half. It's going to involve 2 consecutive cell divisions called meiosis 1 and meiosis 2. And these chromosome cells are going to be duplicated and then, cut in half. So that's why we need 2 stages. So as a brief overview, we can see that, just like every other cell, we're going to start out with a homologous pair of chromosomes from the parent cells. So one chromosome from mom, 1 chromosome from dad. Then we have to duplicate it, okay, to get, the sister chromatids. So remember, one little kind of, little Lego thingy here is what we call a chromatid. So if we duplicate them, we get what's called sister chromatids. So we get 2 of them that are hooked together. Now those 2 sister chromatids in meiosis 1 are going to be divided into 2 separate cells. Okay, and then what we have to do is to make sure that we don't double up on chromosomes in the next organism, we have to divide those sister chromosomes even further to split them apart. Okay, so we're going to get 4 different cells actually at the end of meiosis, and they're each going to have a single sister chromatid that is going to be able to hook up with the homologous chromatids from the other organism in order to make a diploid zygote or diploid offspring. Just like in mitosis, we need to start out with a phase of the cell cycle called interphase, in which the chromosomes are going to duplicate themselves. So that's basically our first step in meiosis, is that we get chromosome duplication. The next stage of meiosis is what we call prophase 1. Okay, so again, it's similar to mitosis, but we have this 1 here because we're going to have, remember we said there's 2 steps to meiosis. We have, the first step, the first phase, and the second phase. So prophase 1 is prophase that occurs in kind of the first stage of meiosis. So in this process, or in this stage of the cell cycle, chromatin are going to start to condense, and special proteins are going to cause the homologous chromosomes to stick together in what are called tetrads. And tetra is, kind of the prefix for 4. And we can see that if we take a look at the sister chromatids, come together, and we've got these little tetrads, or these X's with 4 kind of, two legs for each of them. So if we stick 2 of them together, we get 4 legs. So that's kind of how we get the tetrads here. So we have, 1, 2, 3, 4 little legs here of our tetrad. Now in this arrangement, what can happen is that we can get what's called crossing over, in which these, the different sister chromatids within the homologous chromosomes are going to kind of cross over each other and exchange genetic material. So it's kind of like they twist and break off, and that genetic material is now in on the different sister chromatids. So over here you can kind of see that after it occurred, where you've got some blue chromatids with little red spots and some red chromatids, okay, with blue spots. So that those indicate that genetic material has crossed over and was exchanged between the sister chromatids. So this causes a rearrangement of genetic material and even further, genetic variation among the offspring compared to the parents. As we further prophase 1, those chromosomes are going to condense even more. They'll get more tighter so we can actually see them under a microscope. The spindles, remember like we said in mitosis, these spindles out here are going to start to form. We're going to get those tetrads starting to move to the center of the cell, and our nuclear membrane that's holding all of these chromosomes together is going to start to disappear so that those chromosomes can move to the middle of the cell. So the next phase is metaphase. Okay, so this should be aptly called metaphase 1. And this is where these tetrads are going to align in the middle of the cell, much like mitosis. In mitosis, metaphase is where all of the chromosomes met at the middle of the cell. So the sister chromatids are attached to each other by those centromeres, and the mitotic spindles are going to start to attach themselves to those centromeres. So we can see that here in this metaphase part where we get these little spindles coming out from the centromeres, and they're going to attach themselves to the centromeres in between the sister chromatids. Next up, we have anaphase 1. So again, just like in mitosis. So if you know mitosis, you'll know meiosis. It's just kind of an extra step along the way. So the phases are called the exact same thing. So in this one is anaphase 1. This is where chromosomes are going to start to move to the opposite poles. So we can see that in this one. So those spindle fibers are going to pull the homologous chromosomes away from each other, but those sister chromatids still stay together. So we're going to get, the chromosomes going to opposite poles. So in our final phase of our first stage of meiosis, we get telophase 1 and cytokinesis. And just like in mitosis, this is where the chromosomes end up on opposite sides of the cell. And we get that cleavage furrow, which is going to divide the big cell into 2 daughter cells, with one set of chromosomes in each daughter cell. So at the end of meiosis 1, we get 2 daughter cells here, okay, which now have to have their sister chromatids broken apart so that we can get haploid, the haploid chromatids in more daughter cells, so that when they fuse with the other sex cells of the organism, we get only a diploid zygote, not duplicate, double amount of chromosomes. So meiosis 2 is going to look very similar again to mitosis. So instead of going each phase by phase, basically what happens is that in metaphase we get the meeting of those chromosomes in the middle. Then in prophase 2, the sister chromatids are being attached to the spindle fibers, and in anaphase 2 we get the pulling apart of the sister chromatids. So we get in the end 4 daughter cells with a haploid set of chromosomes. Okay, so these guys will split into 4 daughter cells. If we take a look at the difference between mitosis and meiosis, we will see that they have similar stages, but there are definitely some major differences going on. So the events unique to meiosis are going to occur in the meiosis 1. That's where we're going to see the biggest difference. So if we take a look in mitosis and meiosis, okay, we get that first interphase, which is the duplication of the chromosomes. But then from there, it starts to look a little different. And we can see that in this picture and this picture. So in regular mitosis, those chromosomes are not going to find their homologous pairs. They are just going to end up, lining up in a straight line here in the middle in metaphase. However, in meiosis, we can see that they are going to find their homologous pairs, and they are going to end up meeting in the middle as a tetrad instead of individual chromosomes. Okay, so we have these set of 4 here in the middle instead of lining up just straight in a straight line. So what that does is it's going to give us in mitosis, okay, we are still going to get a diploid cell, okay, with the 23 pairs of chromosomes, right? So we can see in these 2 daughter cells that if we liken the red chromosomes to the ones you get from mom and the blue chromosomes to the one you get from dad, we have a combination of both mom and dad's genetic material or chromosomes. However, if you look at the end of meiosis one, okay, you get 2 cells just like we had, but if you take a look, one of them, okay, is basically going to have one set of genetic material, say from dad, and the other is going to have genetic material, like only one set of genetic material, like say from mom, okay, like the ones that you got from mom. So we get, and this is basically what we call a haploid, because we only have half of the chromosomes in each cell. We've got, the dad's half in one cell and the mom's half in the other cell. So this is basically the biggest difference between mitosis and meiosis. So like I mentioned just a second ago, at the end of meiosis 1, you end up with 2 haploid cells. Now these chromosomes are still duplicated, so we have a double set from mom and a double set from dad in 2 different cells. So now we have to unduplicate those sets, and so that's what meiosis 2 is. It's going to be basically identical stages like mitosis in order to divide the duplicate chromosomes from each other so that we get single chromosomes in each of the daughter cells for, the final mitosis product. So we talked about how the process of meiosis works. There's 2 stages. We have to, double the chromosomes and then, put all the mom's chromosomes in one and dad's chromosomes in another, per se, okay? And then divide those sister chromatids apart so we can get haploid, or half the number of chromosomes in our sperm cells and our egg cells. So let's begin the conversation of how genetic variation comes about. Okay, because the origins of this genetic variation is going to happen in this process of meiosis. So we can basically independent assortment of chromosomes is the origin of genetic variation, and what that means is that we can have 2 times 10 to the 23 possibilities of the different combinations of how the sister chromosomes or homologous chromosomes line up as tetrads, right? This is just 2 possibilities that we have here. And possibility number 1, okay, per se, we could have all of the dad's chromosomes line up on the left and all of the ones that we get from mom line up on the right, right? So we get a purely dad genetic gamete cell and a purely mom genetic gamete cell. And again, I'm saying mom versus dad just to kind of make it a little bit clear on how that works. However, possibility number 2, okay, we could get some of dads on the left and some of moms on the left too, and some of some sets of chromosomes we can get moms on the right or dads on the left, and so we can get here a kind of a combination of these chromosomes in the daughter cells. Okay, so that gives us a lot of genetic variation, okay, of whose genetic material we're going to pass on in what combination to our offspring. So this orientation of homologous pairs meeting as tetrads is basically just a matter of chance, okay? It's kind of complete random chance as to how they actually pair up and who goes to what side of the cell. So we said we get 2 to the 23rd power possibilities because we have 23, remember we have 23 chromosomes, or sets of chromosomes in our cells, so we can have 2 to the 23 power of, possibilities of how those tetrads come together in meiosis 1. That's the first origin of genetic variation. Then we have the fact that fertilization is completely random, right? So 1 out of 8 million sperm that are ejaculated can fertilize 1 out of 8 million cells that are possibly released from the ovaries. Okay, so we get just, again, kind of random chance as to which cell meets which sperm. And then we'd mentioned earlier in meiosis 1 that we can have this crossing over phenomenon occur as well, where, okay, so those tetrads are meeting in, together in the middle of meiosis 1, and sometimes we can get the crossing over those sister chromatids together in this little X kind of shaped region right here. Okay, so this little X-shaped region right here. And we get what's called a chiasma, or site, the site of crossing over, and that genetic information is kind of twisted up or exchanged between the sister chromatids. So we can have complete genetic recombination while, meiosis 1 is occurring. Okay, so we can, exchange some of mom's to dad's chromosomes or dad's to mom's chromosomes, just little chunks here and there. So we're going to get a complete, different arrangement of chromosome and genetic material that way as well. So that's, kind of the origins of our genetic variation and why we don't look exactly like mom and or exactly like dad or have the exact same features as mom or dad. We kind of have a combination of both of them, and sometimes we don't even look like mom or dad because a recessive has been passed down to us that wasn't apparent in mom and dad, so maybe we look like a little bit like grandpa in this way or grandma in some other way. So that's how humans are, basically don't ever look like any other type of human unless you are a twin, okay, a identical twin, and even then they still have different and even then they still can have different features. All right, so unfortunately this meiosis thing doesn't work perfectly every single time. So meiosis can go awry, and this is one of the ways. It's called nondisjunction. Okay, basically either one of the chromosome pairs fails to separate from each other, like here in meiosis 1. Okay, we've got these top two going in opposite directions, which is good. But then if we take a look at this one, okay, both of these pairs are going to one side of the cell. So we're going to get a double pair on, this guy, and then we're going to have a missing one in this cell. Okay, so we're going to end up with, minus 1 chromosome in this cell, and we're going to end up with 3 chromosomes on that cell. So that obviously is incorrect. And then it could also occur in meiosis, I should say, meiosis 2 with the sister chromatids. So in this cell, we had the sister chromatids separate nicely into the 2 daughter cells, so we get, regular haploid cell. Okay, but on the left, these sister chromatids right here failed to separate, so we're going to get 1 egg or sperm that has 3 chromosomes, and then the other ones just has 1 of the little chromatids. So that's going to be a major failure, gone awry. It's going to have dire consequences. Now, most of the time, if this meiosis does go awry and you get, the fertilization of a sperm cell to an egg cell that only has, 1 chromatid of a chromosome, usually, it'll be fatal. That organism actually doesn't develop. But there are some that do, okay? And so some of the results is Down syndrome, okay, which is an extra chromosome in number 21. So they call it trisomy 21 because it's got 3 chromosomes of number 21 versus just the regular 2. So in this case, you can still get the development of that organism, and but as a result, you'll have the Down syndrome, effects. Meiosis usually goes awry with increasing age. Okay, and if we take a look here at this thing, we can also have different conditions occur with abnormal number of sex chromosomes too, not just the other autosomal chromosomes. So if we, if somebody inherits an extra X, it's what we call Klinefelter's syndrome. It's male, but it's got an extra X in it as well. If we have an extra Y, okay, if the Y chromatids don't separate and we have an extra Y, then it actually doesn't really have too much of an effect. It's basically a normal male, so it'll be all right. Now, if we have one of the X chromosomes, or the sister chromatids not separate, we can have what's called a meta female. And if we have one, like if we have an egg that has an X but the sperm didn't get any because the sister chromatids failed to separate and actually give that daughter sperm any of the sex chromosome genetic material, we have what's called Turner syndrome, in which it's female, but they're going to have side effects to that. So that was our discussion of meiosis, and what we're going to do is go on to see how we can get different patterns of inheritance by taking a look at genetics and Punnett squares and Mendel and all that fun stuff that you remember learning from general biology in high school.